Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next generation sequencing competence network that consists of 4 production sites as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
Also from my side, welcome to the NGS-CN Summer School this year on spatial transcriptomics. So as Julia and Daniela already said, this is organized by the German-wide network of sequencing competence centers. This is 4 centers in Tübingen, West Germany, in Kiel, and in Dresden. This year, we in Dresden are the ones heading the organization, bringing together the content, but all of our colleagues from the NGS-CN contribute with content and with organization. So thank you very much, everyone who organized this, and thank you very much everyone to present this. We are here in Dresden with, I counted roughly 40, 50 people. We expect up to 70 people. So we're really happy with the audience here, and we have even more online. I start off with giving a quick brush over sequencing technologies. And that might sound boring for some of you that work already long in science, but actually right now is the perfect time to get an update because there's a lot of movement in the sequencing field. And for everyone who is a little bit earlier in their research career, I think this really gives you a very nice start to see what's coming up in the next 1 or 2 years and gives you a good start, good orientation to educate yourself to the right system that you can use. I start off with a boring slide for all of you. You all know that DNA is actually encoding all the information that's needed for biological life, no matter whether it's human or any other branch of the biological life tree. And DNA is not laying there as plain information. It is actually encoded in a double-strand helix that is very complex, coordinated, and compacted to be stored in the nucleus as a chromosome as we know it. The chromosome is sitting here. This is as a chromatin fiber wrapped around histones that add for its compaction, but also have an impact on the regulation of DNA. And the regulation of DNA actually has an impact on the use of DNA. Usage of DNA I describe as RNA transcription. We come to that in a moment. Today we talk a lot about sequencing. I want to make clear that I talk here about sequencing DNA, okay? Sequencing you can do on many other polymers as well. Today we talk about DNA sequencing. This is a phrasing that I chose. I hope it is correct. Don't take it as 100% gratitude, right? So this is how I grasp sequencing, and this is sequencing is the process of deciphering a repeated structure, in this case a biopolymer, as an unbranched linear chain, and then depicted as symbols so that you can read it. And for biology, this is usually the primary structure of biopolymers. And in DNA, it's actually the nucleotide composition A, T, G, and C. Sequencing by itself, and by this depicting DNA in a readable and rereadable format, for me is actually a quite nice analogy to the Gutenberg printing, where for the first time, writing and storing knowledge was not left for the monks or educated people that were actually able to read and write, but you could actually manifold the knowledge shared amongst the wider public, and with this spread the knowledge and increase the knowledge overall in human life, right? And DNA sequencing is doing exactly that since years now. When I think back to my studies, DNA sequencing was something very new and very expensive, and a few people did it, and it was really only for deciphering the genome, not more. Nowadays, there's a plethora of assays where you can use DNA sequence just as a readout to gain a little bit better a grasp of what's actually happening. And yeah, the use cases are super wide. And I hope I can convince you that the next level of this kind of revolution in using data is coming around the corner. A quick history. I think most of the people thinking about DNA start with this picture, Watson and Crick with the DNA model from the 1950s to 1960s when they developed this, right? But actually, it started much earlier with the Swiss scientist Friedrich Miescher, who actually already in 1869 extracted DNA and described it as nucleic acid, this stuff that actually is the information that is given from one generation to the next. And there's only male pictures here. Nowadays, female contribution to science is now also much better reflected. And in this case, we should not forget Rosalind Franklin with her X-ray studies that allowed to decipher that DNA is actually stored in a double helical structure, right? Without this picture, Watson and Crick would have never come up with the model. Sequencing DNA came later in the 1980s, 1990s, starting from 1975, through Frederick Sanger, who developed a chain termination reaction. In short, it is a PCR where you bring in block polymers that stop the reaction at random places, always either with A, T, G, or C. And initially, this is shown to the left, you would have one lane for G, one for C, one for A, one for T, and you have to run a super long size separation gel to separate then the sequencing. Nowadays, this is done by fluorescence molecules for each base one, so you only need one reaction and you can read 4 signals at the same time. Just as a small hint, this is how DNA sequencing looked in the 1970s, 1980s in the middle, right? Super big gels with a lot of information, and it is a super big hassle to actually decode this. This was the technology that started the Human Genome Project in 1991 up to 2001 with huge parallel machines, PCs, and a lot of machines in one big room using a lot of electrical power to sequencing the human genome. Since then, we have seen not only one, but several revolutions. I updated this just 1 year ago, and already now it's outdated again. I hope I take the newest picture, but we move actually towards a $100 genome. And it started all with a $100 million genome, right? And you see that it is not a linear decrease, but there's actually huge milestones that decrease the cost magnificently. The biggest drop that happened around here was the introduction and marketing, broad range marketing, the idea of shotgun sequencing. And shotgun sequencing is the idea that you do not try to sequence the DNA as a long piece as it is, but actually hack it into smaller pieces, sequence a lot of small pieces. Smaller pieces are easier to sequence, easier to access, easier to amplify. So you can sequence a bigger number of these smaller molecules, and then it's just statistics to stitch them together. And this you do bioinformatically at the end, right? And with this technology, it was actually able to be opening up the whole space of information that is encoded in DNA. I come back to this picture again, not only the linear sequence, but actually the RNA sequence that is copied from it, DNA sequences that are compacted or more open, DNA interaction maps, all of these things you can now assay by use of cheap, massive parallel, millions and millions of reads, short read sequencing, because it gives you the statistical power that you need. Okay, with this, I come to the technologies that are now on the market. And I want to start off with this slide. Quite often, for me as a researcher in a genome center, when we talk about sequencing, people come and say, I want to do Illumina. They don't say, I want to do short read sequencing. They say, I want to do Illumina. It's almost like everyone saying, I will Google something if you just go on the internet and search for something, right? So Illumina spearheaded the fast democratized development of the short read sequencing. And by this, actually got a big market share and almost placed the whole name over the whole field of sequencing. I would like you to double think of this and correct this in the future. Because right now is the phase where Illumina is slowly losing market share. And on top of that, Illumina only stands for short read sequencing. They tried to change, but right now, and in the past, it was short read sequencing. There's more on the plate right now, and this is in a shape that it is not just an outsider option, it becomes relevant. So please try to avoid speaking of Illumina sequencing. try to use short read sequencing. Starting with the first thing, Illumina's patent are running out, and we now have multiple players joining the market for short read sequencing. So all these companies listed there already market devices right now that can do basically the same assays as Illumina can do. And all of them try to find a niche where they are better than Illumina. So you can really think about what you need and choose the right technology for this. All this is short read sequencing. On top of that, next to short read sequencing, you nowadays actually have long read sequencing. Obvious what that is, right? You have high accurate sequencing. I come to this later. Illumina is super accurate, but if this is not enough, there's technologies that are even better. Most of the time, as you can imagine, one of the specialties will cost a little bit more than the average sequencing, right? But if high accuracy is what you need, there's technologies that can address this question. And the other thing is, there's also the possibilities of super fast sequencing. And this is what the whole summer school is about, spatially resolved sequencing. I listed all these blue bots as things that you can do no matter what your assay is. No matter whether you do RNA sequencing, DNA sequencing, chip sequencing, or whatever, you can use all the 4 technologies, long read, short read, super fast sequencing, and decide what your readout will be, right? Spatially resolved is a little bit outside because it is a sequencing technology, but you're actually also restricted to a type of assay that you want to do with it, right? Okay, so let's dive a little bit more into the technology so that you understand what are the differences that are on the market. One is Illumina sequencing. I start with this because, again, this is a spearhead. On this side, I present to you what is actually the principle of how you get to sequencing. You start with DNA fragments. You try to make them short because the sequencer is short. You add connector sequences that allow to immobilize the DNA on a surface. It needs to be immobilized because then you can flush in and flush out chemistry and manipulate the sequence to be able to read it out. It binds to the surface and then it gets amplified on the spot. So that you get clonal amplification locally constricted. Here it says PCR. Nowadays it's actually also isothermal amplification. And the constriction initially happened because you had a pattern flow cell. Nowadays this is an all open flow cell, and it's actually by loading that you separate the molecules and cluster specific spaces. In the end, you dissociate and amplify on the surface so that you get these clusters, as I mentioned. And after you amplified multiple rounds, only then you start to sequence. And you sequence base by base. And this is called flush cycles because you flush in chemistry, you measure, you erase the signal you got, you flush in the next round of chemistry so that you read out base by base by base the fluorescent signal of the cluster. The amplification on the spot is actually quite important. This is needed to get a good enough quality signal because one fluorescence molecule is really hard to measure. You need to collect multiple at the same spot to get a nice signal and quality high enough so that you can find it. And at the same time, base by base reading means you have to wait all the way to the end to read out your information that you want to have. So this technology, and this is listed to the left, gets you really high accuracy, 99.7% base accuracy, billions of reads in parallel because the flow cells allow to bind many, many million molecules. It allows a rather cheap price because it was optimized throughout the last 20 years. The read length is the restriction. It can only go so far up to 300 bases because after that, these flush cycles become inefficient. You get a mixed up signal. So it becomes a little bit bad in quality. And it takes super long. So sequencing takes 2 days here. The machines that allow for the cheap price are actually super high in investment costs, initial investment costs, but Illumina offers a wide range of different machines. So you can also get a cheaper device, but then you have less throughput and actually higher costs per base per information. Okay, with this, I leave, let's say, the baseline Illumina. Let's go to high accuracy sequencing. PacBio, I start with PacBio. They have a small benchtop sequencer that is actually there for high accuracy. They don't do sequencing. Other than that, they do very similar to Illumina, but they don't do sequencing by synthesis, which means the polymerase brings in the fluorescent base. They do sequencing by binding. The only difference is that the base that is built in for the next cycle to come and the base that has the fluorescence to measure the signal are decoupled from each other. So you actually have a blocking molecule, then you bind the fluorescent molecule, you give it time to settle, then you measure, then you flush out the fluorescent molecule, you determinate this first base, and only then you build in. So it brings another round of cycle of bringing in the chemistry, but this allows to decouple the building in of bases with no matter what accuracy to the high accuracy step of binding and measuring the fluorescence. And with this small tweak, you get high accuracy. We don't talk 99.7, we talk 99.9 and higher. Sounds like a marginal increase, but when you think of cancer mutations, you search in 10,000 cells and it's only then 1 or 2 cells. 99.7 to 99.9 makes a huge difference. This allows millions of reads in parallel. It is in the mid-price range. Sequencing up to 200 bases is possible. I already said benchtop sequencer. The investment for the machine and the chemistry is actually higher than Illumina. But this comes for the higher quality. So you have to wonder whether you want to spend the extra money for the extra quality. An alternative to that is Element Biosciences. I moved to that because by principle, it is a mix between PacBio and Illumina and something new. They do not immobilize single-stranded molecules and amplify on the spot. They actually flush in circular molecules, and then they do rolling circle amplification to create a huge ball of DNA of a repetitive sequence. And this they sequence by also sequencing by binding, and the binding is an avidite. This is how they call it. And the avidite is actually a huge blob with a lot of fluorescence sitting there, and then arms out there that have the nucleotide. And the idea is that you flush it in, and the nucleotide binds to the whole ball because at the same space, there's always a space for this nucleotide they are labeled to bind. And then you immobilize this ball of fluorescence intensity. By this, they can, for one, reduce the chemistry cost because fluorescence molecules and DNTPs are super expensive in their use. They can use less concentration because of the higher specificity, and the signal is stronger, resulting in cheaper running costs and high accuracy. This is what they're tailoring for. They have right now already marketed systems in the range of 75 to 300 bases. They have a very versatile sequencer. Also, their machine has more options to sequencing to do. And it allows faster sequencing. The cycle times are faster, so in general, you can do faster sequencing. An alternative to that is MGI. MGI also brings in a circle, but it doesn't immobilize the circle and amplify on the flow cell. Instead, it amplifies it already outside to create nanoballs, and then it flushes the nanoballs in, and the nanoballs themselves get immobilized. After that, they basically do Illumina sequencing, which is fluorescently tagged DNTPs that are built in, and they light up the whole nanoball. So it is a mix of Element Bio, what I presented before, and Illumina sequencing. Again, similar quality to Illumina. They didn't try to get to higher quality, but they're similar quality, billions of reads, faster sequencing again than Illumina, lower price point than Illumina. They can do up to 400 bases in read length. And the machine investment is also lower than Illumina. And also there, they have a huge machine park from small size sequencers to huge machines and can do millions and millions of reads. So they target high throughput, cheap sequencing. And then we have Ultima Genomics. Ultima Genomics is a little bit hard to place because they are, I would say, still actively in development because it's a completely different system. It is not a closed flow cell where you do the flush in, flush out of chemistry. They try to reduce the costs by actually reducing the amount of chemistry that you use by doing the reaction on an open silicon wafer. And the wafer is spinning like a DVD or something like this, right? And then you put a drop of chemistry, and by spinning, you separate it over the whole area in a very thin layer, reducing the cost for the chemistry. And then you do sequencing on the spot, on the different spots on the silicon wafer. It allows even faster sequencing than everything that I presented before, because there you can actually sequence multiple bases at the same time, what they claim. Reduce sequencing cost because of reduction in sequencing material that you use, and that goes up to 300 base pair sequencing. And the idea that they market is also that you can reuse the silicon wafer, being more sustainable for research. You don't trash your stuff. They are still actively in development. They already sell the first machines, but it is not in the normal market, I would say, as of now, at least not in Europe. Okay. With this, I come back to this picture and telling you, don't think of short read sequencing only as one single thing that you can do. Actually, you should think about this sequencer and this sequencing technology that best fits to your purpose, and the purpose on the right side. What do you actually want to sequence and what do you want to study? Okay, now I only talked about short read sequencing so far and high accuracy sequencing actually. We have more players on the market and we're moving away from short read sequencing. Very freshly launched. We have also mid-size read sequencing and high accuracy sequencing just being announced by Roche. It will take 1 or 2 years until they really market their machines. They move away from fluorescent sequencing into electrophoretic sequencing through nanopores. And their main principle is that they do not load dNTPs that are fluorescently labeled, but actually dNTP that has a so-called expandomer on there. And the expandomer is a chemical structure much bigger than the DNA in the dNTP. It's a super long polymer of hydrophobic sequences. And this can actually get stuck and block a pore through a membrane. And by this, influence what electronics are flowing through. And then you can measure it and very specifically say which nucleotide was actually sitting there. They have expanded it by a clever translocation control element, which even more stucks the element in the pore. And you have to actively remove it to move to the next base. And by this, they have full control about the translocation of the molecule and the time they need to read out a high accuracy base read. And this allows very high accuracy sequencing of millions of reads. And because we are not bound to fluorescent sequencing, there's actually 2 advantages, and that is much faster sequencing. You don't need these flush cycles. You can all do this electronically, which is much, much faster than fluorescent labeling and washing in and washing out. And the read length is not restricted by fluorescence intensity blurring. It is actually restricted by the ionic potential over the membrane. So they can go longer. They can go up to 1,500 bases. This is a leap forward compared to the 100 to 200 base pairs that you usually sequence right now. And you maybe immediately see the advantages of doing 1,500 base pair sequencing when you think of RNA, where most of the RNA transcripts are at the range of 1,000 to 2,000 bases and not much bigger. 1,500 bases is actually a very nice spot for biological interrogation. The price tag, however, so far is unknown. So this is really very early access, and it still has to fiddle its way in. But they obviously target something where you need high accuracy and high speed, which is usually medical applications. Okay, now that we made this step away from short reads and we moved already to nanopore, we can go to the first sequencer that actually did nanopore electrophoretic sequencing, which is Oxford Nanopore Technologies. They started their development in 2012 and marketed their devices in 2016, 17. Nowadays, they're, I would say, called established. So the machines are running, they're widely used, and the chemistry is more or less stabilized. What they do is they also have the nanopore, and they also have a membrane through which a potential is running, but they feed the actual DNA through, no expondomer, nothing like this. So very important for me to understand is that this is native DNA or even RNA sequencing. No copy of something or not an expanded chemical molecule. This is really the DNA, if you want. It's the DNA that comes out of your biological sample, right? Speed, as I said, with electric measurement is much faster. Here we talk about 400 bases per second. With Illumina sequencing, one flush cycle for one base takes 2 to 4 minutes. With some extra washes, maybe even 6 minutes. So 400 base pairs per second is a totally different scale. The difference there is with Illumina, you get billions and billions of reads, base by base by base, and each base takes 5 minutes. With long read sequencing, you get 400 bases per second, but you only get 400 molecules at the time. So you have to wait until the first 400 molecules are done before the second 400 molecules are loaded, and so on and so forth, right? But this is a massive amount of speed faster. Also, the read length is not restricted. Actually, the record right now is somewhere between 4 and 5 million bases. One read, one native read. 100,000 to 200,000 is no problem. 1 million is now often seen. 4 or 5 million is the record right now. And the restriction there is actually not the chemistry and the device, it is really more sample preparation. So this is one of the last points. You actually need to make a very nice sample before it's worth going on to Oxford Nanopore sequencer. It's also very expensive to run. Another advantage is that the sequencer became much smaller. You don't need a microscope around it and stabilizing it for perfect measurement. It is actually an electronic device, so that you can have a handheld device that can sequence millions and millions of reads. And the first reads, the first sequences are done after 10, 15 minutes. And this opens a new field of sequencing. So this device was actually sequencing in space at the ISS. It was in the Antarctic, in the jungle, orang-utans. So this is really also a difference in scale of machine. Coming to the last sequencer, this is long read sequencing PacBio. I mentioned PacBio is a short read sequencing. PacBio started as a long read sequencing. They want to become now the whole board and have short read sequencer, midsize and long sequencers and high accuracy sequencers. They can do also long read sequencing. The sequencing is not as long as Oxford Nanopore, to millions of bases. The technical limit is 150 kilobases, and they usually sequence with 15 kilobases, but focus on super high quality. 15 kilobases is still a big deal different than short read sequencing, right? So it's a different beast. And you only get millions of reads, not billions of reads. It's also more expensive, but the accuracy is also super, super high. And if you want also here, you can sequence native DNA without amplification. What I did not mention with Nanopore, it's true for Nanopore and PacBio, you can sequence DNA methylation without any chemical interrogation, you just read it out. The principle is here, kind of like Illumina, you sequence by measuring fluorescence when it is built in the sequence, but while in Illumina you immobilize the DNA, what you immobilize here is actually the enzyme that brings in the next base. And you immobilize it at the surface of a glass slide, and you light by total internal reflection microscopy from the bottom. So you only measure something that really sits in the enzyme. And by this, they reach high accuracy, live sequencing. And the super high accuracy, which is much better than Oxford Nanopore, comes from reading the same DNA molecule multiple times because you have a circular DNA molecule. So you read basically every base 5 to 10 or even 15 times and make an average out of all these measurements, increasing the specificity. Okay, with this, we have the quick brush of sequencing technologies done. I come back to this slide telling you, you should really think of what you want to do, what you want to address, and then choose the technology and not just think of sequencing as sequencing. It is not short read sequencing anymore. You can decide yourself what you really want to do and then choose the technology that fits best. And as one example that you can do for sequencing, I want to highlight RNA sequencing. I stole some slides from a very nice GitHub repository where there's very nicely described what RNA sequencing means, just as a memory. A DNA encodes the sequence, but it's actually RNA that gets transcribed from it, spliced, and then brought into proteins, and the proteins make the function of the cell and other functional units. So if you assess what RNA is expressed at which state, you can describe the state of each cell and decipher what cell we're talking about. Also, each gene can use different pieces of its information to make different proteins. So to understand the RNA actually gives a very nice description of the cell itself. The way to get there is that you extract your RNA, you make a copy of you extract your RNA, you make a copy of it in cDNA. This you hack into pieces and do short read sequencing or long read sequencing or midsize sequencing. And then you use this information to map it bioinformatically back to the known information and annotation before, which gives you then a description of the state in form of grouping of different samples or different states and finding out which genes are important. This is a test data set of cars and different features to cars, but the principle is exactly the same if you want to study biological samples. Okay, this is RNA sequencing. Keep that in mind because from here we go on away from the technology into the applications, single cell sequencing, spatial sequencing. Most of these address RNA sequencing as the foundation. And with this, I'm at the end and would like to leave the rest of the school to spatial sequencing. And I'm open for questions. Thanks.